This is the actual unit that we have already begun. External morphology of uni or bivalve. And these are the points which I have already discussed. Introduction, systematic position, habitats and habits, external morphology, which includes shell, how it appears externally, internally, and the mental cavity. The introductory part, it is a freshwater mussel found buried in the waters of Indian lakes, ponds, and rivers. It is edible, it's scavenger, it keeps the water clean, and it has some commercial value. The shells are very important, especially these they are used, the powder of the uh, shells, that is, as it is rich in the calcium carbonate, it is used in the poultry feed to provide nutrients. Systematic position, we have discussed on the systematic position, where it lies in the animal kingdom. It belongs to phylum Mollusca, whereas it is soft bodied. It belongs to class Pelicypoda. The foot is present, triangular, muscular, shear like foot is there. It is bivalve. Two shells are present, because you know the snails are there which are having only single shell, which is also twisted. But here two equal sized valves are present. It belongs to order Eulamelibranchiata. Interlamellar junction, W-shaped gills are there. Gills are provided with the cilia, and the cilia, they play important role in creating the water current which enters into the body. It also leaves the body. It belongs to family Unionidae. We two equal sized adductor muscles. Now these are the muscles. Set of muscles are there which play important role in closing and opening the shell. Genus Lamellidens, plate-like gills, and species Marginalis. Habitat. As I have already mentioned, that it is found in river, lakes, ponds, and found buried in the upper layer of sand or at the body bottom. When buried, the posterior tip of the shell remains exposed. It's out of the soil or out of the mud to allow the entry and exit of the water. Habits. It maintains the water current by the ciliary action. The cilia are present on the gills. It is omnivorous. Its food appears in the form of microorganism of both origin of plant as well as animal as well as plant origin. It feeds by filter feeding mechanism. It creeps. It is sluggish. It moves very slowly. See, it covers about 16 kilometers in a year. Foot is used for burrowing. So the locomotion and the burrowing are the functions carried out by the foot. It moves in shallow water at night and returns at the deep during day. Sexes are separate. Development sexes are separate, means male and female are separate, but you cannot make out whether the animal is male or female externally, because it do not exhibit any sexual dimorphic character. If disturbed, it withdraws foot and siphon inside the body and close the shell tightly. See, this is how the animal appears. This is a bivalve, गोड्या पाण्यातला शिंपला, what we call it in Marathi, शिंपला. How it appears, morphological features. Now, first of all, we will discuss on the shell, its external feature, how it appears. And these are the different points: shell, hinge line, what is meant by umbo, lines of growth, foot, and siphons, that we are going to study in details, but just we are going to touch highlight. The shell, as I have already mentioned, shell, two equal valves are present. This is the diagram from the book, and this is the diagram which I have sketched for it. So this shell is there, hinge line. This is the border. This is the entire border which is called as hinge line. See, this is the border which is called as hinge line, and this structure, hinge ligament, this is the tissue which holds hinge शब्दशः मराठीत अर्थ आहे हिंजचा बिजागिरी. जशी दाराला बिजागिरी असते, like that it is a sort of a device which holds the two valves together. So hinge ligament is there keeping the two valves together. Umbo, this is the part, umbo, whitish part of the body, bulged part rather you can say, and this is the part which is grown first during the embryonic condition, and the lines of growth, these are the lines, you can see the concentric lines are added as the animal grows, so these are called as lines of growth. From these lines of growth we can calculate the age of the foot. Now, here is the location of the foot when the animal is active. But once it is disturbed, both the structure, foot as well as the siphons, they are withdrawn in. So, location of the foot is at the anterior side, while as the location of the siphons, both inhalent and exhalent siphon, they are at the posterior end. Both the structure, foot as well as these exhalent and inhalent siphons, they are withdrawn when the animal is disturbed. But when it is fearless, when it is moving, when it is in a favorable condition, it withdraws its foot, or it protrudes its foot out and undergoes the sluggish locomotion, as well as it keeps these two openings open in order to allow the entry. So entry of water in the body is through this inhalant siphon, and the exit of the water from the body is through this exhalent siphon. Both of them are present at the posterior side. Shell, how it appears internally when the animal is removed. Especially, the shell shows different types of scars. Run. Scars, these scars are nothing but these are the scars of the two sets of muscles, anterior muscles and posterior muscles. See, in anterior muscle, there are three sets of muscles: anterior adductor muscle, anterior retractor muscle, and anterior protractor muscle. While in case of protractor muscle, there are only two muscles: posterior adductor and posterior retractor muscle. See, this is the diagrammatic view after the removal of the animal. This shell, how this right shell appears internally. See, so this is the scar of the anterior adductor muscle. This is the scar of posterior adductor muscle. Scar of anterior retractor muscle, scar of anterior protractor muscle, and this is scar of posterior retractor muscle. So both these muscles, they play a role in opening and closing of the shell. Now, another structure is a pallial line. This is the line which is running from, which is running from the anterior adductor muscle up to the posterior adductor muscle. This is the demarcation. This is the line which appears internally on the shell, and this is the pallial line. Now, this line indicates the limitation of the mantle lobe, and mantle lobes is nothing but it is the internal lining of both these shells. So, the mantle lobe do not exceeds beyond the limit of this pallial line. Hinge teeth. This is the anterior tooth, hinge tooth, and this is the posterior hinge tooth. Both these tooth, when the two shells are closed tightly, these two tooth of both the shells, they entangle with each other and hold the shell very tightly. So this is how the shell appears internally when the animal is removed. Now, this was the point which I think Nikita has missed last time, and that's why it's a revision for her, and it is also an introductory part to all of you, rest of the students. Mantle, we are going to study mantle lobes and the mantle cavity. See, this is the diagrammatic structure, and this is how the animal appears inside the shell. How it appears. Now, in this diagram, you will find what are these mental lobes? Mental lobes are, let's say, whatever which is shown in spots. Entire this is the right mental lobe, and this is left mental lobe. Now, left shell has been removed, keeping the mental lobe inside, and here just the left mental lobe is lifted up so that to visualize the internal organs. So what organs are now which are visualized here after lifting up the mental lobe? These are nothing but foot. This is the foot which is already withdrawn in. Labial palps, which are also referred as the lips of the bivalve, which surrounds the mouth. This is the mouth. This is the location of the mouth, and the gills. See, these are the gills. Outer gill lamina, which is smaller. And this is the inner gill lamina, which is larger. Okay. So this is the structure. On at the posterior side, as I have already mentioned, that is exhalensiform, inhalensiform, which are already withdrawn in. This is the location of the hinge ligament. Location of the anterior set of muscles, posterior set of muscles. And see, this is the anterior muscle, this is the posterior. Actually, in the live condition, how this is the lobes, so this is the gills, these are the labial palps, foot, and the mental lobes. So, how this right and left shell appears? Internal structure, scar of anterior adductor muscle, scar of posterior adductor muscle. This is the tooth, anterior, this is the posterior tooth, this is the palatal hinge line, and the hinge ligament. Okay. So which these are, so this is which already we have discussed. Now I am going to share another. Now this is the topic which we are going to. Can you see the screen?
Yes, sir. Yes.
Okay. Now we are actually beginning with another unit. Today's unit, that is the functional anatomy and functional anatomy of bivalve. We are going to continue. So digestive system of bivalve. This we are going to study in details. So in this topic, we are going to study following points. These are alimentary canal, which consists of mouth, esophagus, stomach, intestine, rectum, and anus. Second point that we are going to study is the digestive glands. And the third major point that we are going to study is the physiology of digestion. So these are the different points which we are going to cover in this today's lecture. So let's begin with first alimentary canal. Now, before going into details with the digestive system, and while studying the digestive system, one should know why the digestive system is studied, and you should know the simple meaning of the process of digestion. Can you tell me, can we define what do you mean by digestion? Yes. Can you tell me the simple definition of digestion? Anybody? Yes? Can you hear me? Hello?
Yes, sir. The
Oh.
food we can to the mouth
Huh?
for the further. We need a protein, vitamins for our growth. We need a food.
Uh-huh.
Sir.
Yes.
मग डायजेस्टिव्ह सिस्टीम थ्रूच आपण ते घेऊ शकतो. जे काही आपल्याला फूड थ्रू हवंय ते डायजेस्टिव्ह सिस्टीम थ्रूच घेऊ शकतो.
Okay. Anybody else can correct this in more technical words? Yes. You are right. but I want a definition which should be uh, suit the students of BSc Part 3. Yes. Digestion is nothing but conversion of, yes, come on, help me to complete the definition. Hello, Nikita, are you there?
Yes, sir.
Conversion of
Hello.
complex food which is in the form of carbohydrates, proteins, and fats into absorbable form. काय होतं कार्बोहायड्रेटचं कन्व्हर्जन कशात होतं? Carbohydrates are converted into this is the complex, the carbohydrate, proteins, and fat. This is the complex form of food, isn't it?
Yes.
It
Simple
should
form of sugar.
it should be converted into simple, digestible, absorbable
Simplest
form.
form of sugar.
So my question is, carbohydrates are converted into what? They are converted into simple sugars. Like for example, glucose.
Yes.
Proteins are converted into
Maltose.
proteins are converted into amino acids, and fats are converted into fatty acids and glycerols. Okay.
Okay.
Now, mouth. As I have shown you the location, mouth, it it lies at the anterior end of the visceral mass. You have seen this mouth in the diagram. Still further, we are going to study. See, this is the location of the mouth. Now, this is the diagrammatic view. Can you see the location of the mouth on the screen? Here is the location of the mouth. Say yes or no. Can you see this? Hello? Okay.
Yes, sir.
So mouth is there. Location just below the posterior border of the anterior adductor muscle. See the location. It is bounded by on either side by two fleshy lobes, labial palps, which I have shown you. Now see the labial palps, their outer palp of the two sides. आपण एकाच बाजूचे labial palps बघितलेले. Outer palp of the two sides unite above the mouth to form upper lip, and the inner palp unite to form lower lip. So mouth is surrounded by upper and lower lips formed by the labial palps. The outer surface of pulp is heavily ciliated, which helps in driving the food in. And very important to note is that jaws, radula are absent. Now these are the structure where these structures are present in mollusk. In which mollusk you have already studied these two types of structures which are present in the mollusk last year. Which mollusk you have studied last year? As a type study, पायला शिकला ना पायला. Have you studied पायला? In pila, jaws and radula are absent. Sorry, they are present in pila, but they are absent in bivalve. So mouth opens into short tube called as oesophagus. It is a very short, extend backward, upward and backward, and opens into stomach. Now, actually, you will find the see. This is the oesophagus, which runs upward and opens into the stomach. This is the location of the stomach. Okay. See, location of the stomach. Now, stomach. It is wide ovoid sac surrounded by the digestive gland. See, this is the digestive gland which is surrounded. Near posterior side, it gives off ventrally a tubular diverticulum. Now, this structure is very much important because it is playing an important role. Diverticulum called style sac. The style sac contains gelatinous rod called as crystalline rod. Now, see, yes, this is the diagram which I have sketched. See, location, esophagus is there, stomach is there, digestive gland is there. This is the portion. This is called as style sac, and this is the enlarged part of this. This is the portion entire which is shown here. Style sac. Inside this is the rod-like structure called as style. Crystalline style is present, and see, the rod is made up of concentric layers of mucoproteins secreted by the ciliated epithelium of sac, and that contains the enzyme amylase and glycogenase. Important, very important. The structure is typical present in case of this bivalve. The upper end of the style projects into stomach to meet the hard chitinous plate called as gastric sheath. Now see, this is that gastric sheath, and this lining, this wall, contains the epithelium cells which are responsible for secretion of the enzyme. This is the hard chitinous part present at the roof of the stomach called as gastric sheath. See. So, the cilia of the style sac rotate the style so that the upper end rubs, strikes against the gastric sheath, and the fragments containing enzyme breaks off from it and gets mixed with the food. Now see, means during the course this rod moves in the twisted form and it often strikes, so often strikes, and the fragments are released, tukde tukde padtat, and these are the broken fragments are nothing but these are the having the enzyme inside it. Next part is intestine. So it starts from the floor of the stomach in front of the style. It first descends khali utarte into the upper part of the foot where it gets coiled upon itself and then descends parallel to the descending part at the level of the pericardium and bends backward and continues as a rectum. See how it appears. So this is the intestine. It starts from the descends down from the stomach in front of the style. Style pude, and it descends down like this. Foot parante khali yete. Again, it gets again it descends down. Sorry, down. Again it becomes coiled. It ascends and it moves here, enters. So this is the portion. This is the location which is called as pericardium. This is the chamber which is called as pericardium. So pericardium म्हटलं की काय? It lodges the heart inside it. See, this is the location of the heart. Ventricle is there, auricle is there. We are going to study it in details while studying the circulatory system. But just to keep in your mind that the intestine descends down, coils here, and around this, these are the gonads, reproductive structures. It is coiled here. Again, it runs upward and enters into the pericardium. See, and continues as a rectum. Now, rectum. It runs straight backward through pericardium. म्हणजे pericardium मधून pass होऊन जाते, where ventricle of the heart surrounds it. After leaving the pericardium, it continues up to anus. The wall of the rectum is produced internally in a longitudinal midventral fold called typhlosome. Now, the word typhlosome, where you have come across this word typhlosome, you have come across this word typhlosome in your BSc Part 1 class while studying the earthworm. Do you remember where this typhlosome is present in the earthworm? Of course, it is present in the earthworm. See, we are studying, we have already studied earthworm as a non-chordate animal. Now, at present, we are studying the animal which is also a non-chordate. That earthworm is an annelid, and here we are studying the mollusk. In case of earthworm, you'll find that the typhlozoa is present in the intestine, but its location is at the dorsal side. Dorsal typhlosolar intestine. The intestine which bears this typhlozoa is the typhlosolar intestine. Pre-typhlosolar, typhlosolar, and post-typhlosolar intestine. These are the three parts of intestine in which the intestine is divided in case of earthworm. But here, in case of bivalve, the typhlosoal is present, which is the mid-ventral fold, completely opposite. Mid-dorsal in case of earthworm and mid-ventral in case of this. It was present in case of earthworm in the intestine, but in case of bivalve, it is present in the rectum. So this is the difference. Then, so see, so this is the intestine which is passing through the ventricle, and it is emerging out of this pericardium and continues as a rectum. Okay, it continues as a rectum, and it ends in the form of anus. Now this anus, small opening situated in the posterior part of, आता इथे शब्द बघा, suprabranchial chamber. शब्द काय वापरलाय? Suprabranchial chamber on the dorsal side of exhalent siphon, serving as cloaca. Spelling cloaca, serving as cloaca. Now see, this is the exhalent siphon. This is the area which is referred as cloaca. This is the anal opening, terminal part of the rectum. So, why this area is called as cloaca? Because not only it receives the undigested food which is egested out through the anus, but it also receives the reproductive gametes, you can say, sperm and the ova, which are also received in the cloaca, which are released out through the exhalent siphon. Also, the excretory products which are formed through the excretory system, that are also brought into the cloaca, and then they are removed or given out through this excellent siphon, so referred as cloaca. So it is opens here, and along with the outgoing water, the undigested food particles are there removed. Now, word suprabranchial chamber is used here. See, while studying the mantle cavity, we have come across the word gill lamina. Try to recollect the location of the gills. Just now I have shown you the mantle cavity. Now above, see how the word is used here, supra. Supra means to the dorsal side, above. Branchial, the word branchial is referred to gills. Okay, so roughly you'll find the gills, the location of the gills is here, and the inner opening is here at the above, at the dorsal side, in the suprabranchial chamber. So that's why the word is used as suprabranchial chamber. Anus opens in the suprabranchial chambers in cloaca. Is that clear? So digestive system, I repeat, mouth, a small opening is present which is surrounded by upper and lower lips. Upper lip, बघा आपण इकडे एकाच बाजूच बघतोय. This is the labial palm, outer labial palm. This is the inner labial palm. Its corresponding is the here. So here will be the another part of the outer. It is hidden, you can say. So it forms the upper lip, the counterpart of the inner labial forms the lower lip here, which surrounds the, and these labial palms, they are highly ciliated. These cilia which are present on these labial palms, they also select the fine particles of the food, and they allow the food particles to enter in, or they are, if they are not suitable, they are rejected and they are dropped in the mantle cavity. So here this in mantle cavity म्हणजे इथं ते ड्रॉप केलं जातं. Okay, mouth followed by esophagus, a short tube, followed by stomach, oval structure, surrounded by digestive gland. Posteriorly, it has a outgrowth called as style sac. This style sac is glandular, you can say. Its epithelium secretes the enzyme. The style sac holds the style, crystalline style, a rod-like structure inside it. And this rod protrudes, enters into the stomach. Posteriorly and the anterior-posterior side of the stomach, here is the hard chitinous structure called as gastric shield. So this is the location of the gastric shield. The cilia which are present in the epithelium, ciliated epithelium is the tissue which is present in the wall of this sac. The movement of this cilia makes the rod rotate, and movement of this rod is done so that it strikes against the shield and it releases the fragments which contains the enzymes which gets mixed with the food, that is in the whatever food is taken in. Stomach is followed by intestine. So intestine descends from the stomach. First it runs down into the gonads, then it ascends, it descends, it gets coiled, again it ascends upward, runs parallel to this descending limb, parallel, and then it enters into the pericardium. Pericardium lodges, or it holds the heart, and through the ventricle, passes through the ventricle. This is a very unique feature of the bivalve. In no other animals you will find the intestine is passing through the ventricle, but in case of this you will find it is passing through the ventricle. It emerges out of the pericardium and continues as a rectum, a small tube. Rectum ends in the form of anus opening with a sphincter. This opening, it opens into the cloaca. And this opening, as I mentioned, is in the suprabranchial chamber, which is in contact with the exhalent siphon. Exhalent siphon plays important role in, or it is for the exit of the water. So suprabranchial chamber ची लोकेशन आहे आणि हे इनहेलंट सिफॉन इथं आहे इन्फ्राब्रेंचियल चेंबर. म्हणजे इथं रिजेक्ट केलेले फूड पार्टिकल्स आहेत, हे काय करतात, ह्या परत इथून वर ढकलून ह्या सुप्रामधून ते परत बाहेर फेकले जातात. ओके. So this is the style sac in a larger view, esophagus, stomach, which I explained to you. Now the digestive gland. Second part, digestive gland. You have seen the location of the stomach, and stomach is surrounded by the digestive glands. It is a diffused gland, always found surrounded above the stomach. They are in pair, irregular, dark brown or green masses on the sides of the stomach. It opens in the back. Now, very important structure is that it opens into enterodorsal part of stomach by many ducts. Digestive glands, even though it is surrounding the stomach, it is opening in the enterodorsal, stomachच्या वरच्या बाजूला, म्हणजे इथं याच्या अवतीभोवती काय सगळं डायजेस्टिव्ह ग्लँड आहे. and enterodorsal side, this is the location where you'll find these ducts of these glands. They are opening into the stomach. The duct shows two tracts: non-ciliated inhalant tract for carrying food particles into the gland, and ciliated exhalant tract for carrying waste back into the stomach. The gland not only secretes digestive gland, but itself ingest the fine particles to bring about intracellular digestion. See, gland is there, non-ciliated tract म्हणजे स्टमक मध्ये आलेलं अन्न हे डायरेक्ट त्या ग्लँड मध्ये जाते आतमध्ये. इथून आतमध्ये डक्ट्स आहेत, त्या नॉन सिलिएटेड आहेत. फूड एंटर्स into the gland, आणि दुसऱ्या टाईपची डक्ट आहेत की सिलिएटेड डक्ट आहे की जे then this digested food digestion takes place, and undigested food is brought back into the stomach by the ciliated tract of the duct of the digestive gland. So two types of ducts are there: the duct with ciliated tract and ducts without ciliated tract. So, and the ducts, as the ducts are surrounded by the cells, epithelial cells, and these cells also play role in the intracellular digestion. Means these cells are having the capacity to engulf the food particles. When the cell has the capacity to engulf the food, then those cells are called as, there is a special term in the cytology where this the cells which engulf the food particles, that is called as, do you know the meaning? Do you know the exact word for that? Hello? Hello? Answer me, please. Hello? 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 Nikita?
Hello, sir.
Can you hear me?
Yes, sir. Can you repeat question?
Yes. The cell which has a capacity to engulf the food particles, those cells are called as dash. What those cells are called as?
mitochondrial cell.
No. Now amoeba is a single cell, isn't it?
Yes.
And amoeba feeds by taking the food inside the cell, right?
Right, sir.
So what that mechanism is called as?
Egestion.
Egestion. Well, very good, egestion. But that process that, now consider amoeba as a cell, not as an animal, as a cell. So such behavior of the cell to engulf the food particle, that is called as phagocytic cell. Are you getting me?
Yes, sir.
Phagocytic cell. And this epithelium of these ducts, they are having such phagocytic cells, and which ingest the fine particles to bring about the intracellular digestion. Is that clear? Okay. Now physiology
Oh,
of
sir.
digestion. This is the third part, that is physiology of digestion. Now it is often studied under food, feeding, how the animal feeds, digestion, absorption, assimilation, and the process of egestion. Okay. Now food. It is a planktonic feeder. Food consists of diatom. Diatom is an algae, protozoans, animal. Means, as I have already mentioned earlier, that is, it is omnivorous in diet, but its food contains of the microscopic organism and the organic detritus part. Because as it is found buried, you'll find the organic detritus which settles at the bottom of the pond or lake, or all that organic matter. When it disintegrates, these are converted into fine particles, and those fine particles enter along with the incurrent circulating water into the body. So this is the food, and as it is feeding on the planktons, microscopic organisms, it is called as planktonic feeder. Feeding mechanism. It is a filter feeder. Now, here gills are very much important. They are playing important role in the feeding mechanism. Filter, गाळून घेणे, straining food from large quantity of water. For this purpose, the cilia which are present on the gills, they are playing an important role. Now, what are these lateral cilia, frontal cilia, that we are going to study while studying the gills and structure of the gills. Just now you keep in your mind that certain special cilia present on the gills, they are playing important role in the filter feeding. So lateral cilia of the gill filament and cilia covering the inner surface of the mantle. The mantle, you have already known that mantle is nothing but it is the covering inside आतलं lining. थोडक्यात शेलच्या आतमध्ये असलेलं lining is called as mantle or mantle lobe. Draws water current in the mantle cavity. Now water current कुठून येणार? Through the inhalent siphon. कुठे येणार? तर infrabranchial chamber. Now where this infrabranchial chamber is there? See, we'll go back and I will tell you. See, this is inhalent siphon, and here entire area, this entire area is infra. कारण इथं लोकेशन कशाची असते? गिल्सची लोकेशन. गिल्सच्या खाली इन्फ्रा, गिल्सच्या वर सुप्रा. एनस कुठं आहे? सुप्राब्रँचियल चेंबर. इथली लोकेशन आहे बघा, इनहेलंट सिफॉन, पाणी इथून आत येत आहे आणि पहिलं कुठं जाणार आहे? इन्फ्राब्रँचियल चेंबर. ओके, so it enters into the infrabranchial chamber through inhalant siphon. This current brings food particles. On entering into mantle cavity, the heavier, तिथंच जड जड जे काही particles असतील, sand grains असतील, ते तिथंच राहतात. They sink down at the bottom of the mantle cavity, where cilia drives them out between the mantle lobes near the posterior end. म्हणजे परत suprabranchial chamber कडे त्यांना ढकललं जातं जेणेकरून so that they can go out along with the outgoing water current. But the lighter particles pass over the outer faces of the gill filament where they entangled with the mucus. आता म्युकस बरोबर त्याच्या चिकट पदाबरोबर ते फाईन पार्टिकल सगळे मिक्स होतात. फूड लॅडन म्युकस, आता म्युकस मध्ये अडकलेलं अन्न, फूड लॅडन म्युकस masses are sent ventrally by beating of the frontal cilia of the gill filament to the food groove of the lower edge of the gill lamina. म्हणजे लॅमिना गिलची जी बॉर्डर आहे त्या बॉर्डरकडे हे सगळे अन्न पदार्थ अन्नकण ढकलले जातात, त्याची एक फिलामेंट तयार करून, कॉर्ड तयार करून. ते सिलिया फूड ग्रुप म्हणजे ते एक त्या गिल लॅमिना अशी खाच असते की ज्याच्यामध्ये ते अन्न जाऊन बसतं. आणि तिथले जे सिले द सिलिया ऑफ द फूड ग्रुप डायरेक्ट द फूड मासेस फॉरवर्ड म्हणजे कुठं, आता बघा, दे रीच द सिलिएटेड ग्रुप प्रेझेंट बिटवीन द लॅबियल पाल्म्स, म्हणजे तोंडाकडे आता वाटचाल चालू आहे. Labial pass to reach the side and direct the food masses into the mouth. Labial palps are also ciliated. मगाशी सांगितलं मी की labial palps are also ciliated, and this cilia now helps the food, or the cilia directs this food towards the mouth. The sorting of food takes place at the labial palm. आता मगाशी मी सांगितलं की सेन्सरी, जर वेदर इट इज पॅलेटेबल ऑर नॉट, वेदर इट इज सुटेबल ऑर नॉट, वेदर इट इज फाईन ऑर नॉट, ओनली दॅट फूड इज टेकन अगेन allowed to enter into the mouth, and the heavier food masses are dropped to the bottom, to the mental cavity, and are eliminated. Through mouth, food passes into the esophagus and then into the stomach. Okay. आता process digestion, masticatory organs मगाशी सांगितल्याप्रमाणे jaws, radula are absent. No need to masticate because only fine food particles are allowed to enter into the stomach. In the stomach, आता now the role of the style sac. Crystalline style acts as the stirring, अन्न घुसळण्याचं, ढवळण्याचं काम that crystalline rod is doing. Its rotation by style sac causes the detachment of the food particles from the food masses. जी अन्नाची साखळी तयार झालेली असते त्या साखळीचे परत तुकडे the string is broken with the help of this rotation of this rod when it strikes to the gastric shield which is present on the dorsal side. And while doing this style releases the enzyme into the stomach which hydrolyzes the carbohydrates extracellularly, म्हणजे स्टमकच्या कॅव्हिटी मध्ये. Then fine particles of food are passed into the digestive glands. बघा, नॉन ग्लँड्युलर डक्ट, परत स्टमक मधून स्टमकच्या भोवती असलेली सराउंडिंग द डायजेस्टिव्ह ग्लँड इज सराउंडिंग द स्टमक, but the ducts are there which carry the food particles into the gland through inhalant ducts which are non-cellular, where it is engulfed by the phagocytic cells. मगाशी मी सांगितल्याप्रमाणे म्हणजेच काय करते तिथं, इंट्रासेल्युलर डायजेशन ऑफ कार्बोहायड्रेट्स, प्रोटीन्स आणि फॅट्स with the help of enzyme takes place. So process of digestion is completed. Absorption. The digested food diffuses from the gland into the blood circulation because blood capillaries are supplied to the directly तिथून blood. Assimilation, process in which the absorbed food is taken by the body cells and used for energy growth and repair is called as assimilation. Egestion, important part. Egestion. Now, whatever food is undigested, that undigested food particles are sent back into the stomach. बघा, परत आता कोण काम करतंय? Ciliated ducts of the digestive gland send the undigested food into stomach. From there, it passes into intestine. It is converted into fecal pellets, बारीक बारीक गोळ्या. pellets and then passed into the rectum. From the rectum, the feces is passed through the anus into the cloaca to be carried out with the outgoing water current, that is excellent siphon. Means again you will go back. Yes. See. So now undigested food is coming here. Again it is passing through the intestine. Intestine मधून असं पास होऊन परत ते रेक्टम मध्ये होतं. पिलेट्स तयार होतात. इथं परत एनस मधून क्लोएका मध्ये फेकलं जातं आणि ह्या बाहेर जाणाऱ्या पाण्याबरोबर through this outgoing water the undigested food is thrown out and that process is called as egestion. So here the physiology of digestion is completed. So physiology of digestion is always studied under the headings such as food, feeding, digestion, absorption, assimilation and egestion. So we have completed this topic, digestive system of Unio, under three main headings: alimentary canal, digestive glands and physiology of digestion. So here we have completed the topic. Now let's have a discussion. If you have any queries, if you have any questions, I'm ready to solve your queries. So let's have a discussion. Do you have any questions?
No, sir.
Have you understood?
Yes, sir. Sir,
Yes.
नोट्स काढ PDF भेटेल का अजून थोडंसं डिटेलिंग साठी?
हो.
सर, हां.
मिळतील.
हो सर.
So I hope the time is up, right?
हो सर.
So shall I stop now?
Yes, sir.
Okay. Stay home, stay safe. Yes.
क्वेश्चन होतं.
Huh?
ज्यांचे फिशरीत ऍडमिशन झाले, जर बॅच कमी असेल तर कोलॅबोरेशन नाहीत ना करणार ते झुलॉजी मध्ये?
नाही. तुम्ही जे आता एक दुसरं एक आय वॉन्ट टू टेल यू दॅट, आता तुम्ही झुलॉजीचे लेक्चर अटेंड करताय की नाही?
हो सर.
ते पहिल्या लेक्चरला मी सांगितलं की जो सिलेबस आहे मॉलिक्युलर बायोलॉजीचा, तो कॉमन पेपर आहे बरं का.
येस सर.
तो झुलॉजी मध्ये पण आहे आणि फिशरी मध्ये पण आहे.
हो सर.
आणि बायोस्टॅटचा पेपर तो तिकडं पण आहे आणि इकडं पण आहे. त्यामुळे ते लेक्चर मिस करू नका झुलॉजीचे, तुम्ही कॉमनचे.
झुलॉजीचे पण
इवन एस्पेशली just a minute. Okay, now I will stop the sharing, but now we'll have the discussion.